Hello everybody, welcome to the Enabling and Optimizing High-Quality Spectral Flow Cytometry Analysis, the Cytek Aurora and FCS Express Synergistic Solution webinar. On today's webinar, we have myself, Sean Burke from Novo Software, and we have Janelle Shook from Cytek Biosciences. We're going to be presenting on some of the technology and techniques involved with spectral flow cytometry data analysis and the solutions that are offered in FCS Express. So you may have heard by now that Cytek Biosciences and Novo Software have partnered to seamlessly read raw spectral data in FCS Express. And this collaboration brings the powerful combination of cutting-edge spectral flow cytometry technology from Cytek together with the advanced data analysis tools in FCS Express. Additionally, Cytek systems purchased after July 6, 2018 are eligible to receive a license of FCS Express. So if you'd like to get involved with that, we encourage you to reach out to your representatives from Cytek, ask them about getting that license of FCS Express. They'll be able to help you out. On today's webinar, we're going to focus on pairing Cytek spectral flow cytometry technology with FCS Express to acquire quality data and how downstream tools in FCS Express can facilitate further analysis, quality control, and reporting of those results. So to start the webinar today, I'd like to introduce Janelle Shook. Janelle, are you there?
Yes.
Great. We're going to make Janelle the presenter here. She's going to give you the first portion of this webinar about some of the technologies from Cytek Biosciences. All right, I'll let you take.
Okay, thank you, Sean. So to get things started, we can't get to all of Sean's exciting tools that he's going to demo for you in the FCS Express software until we have some great data to feed into it. So to start things off, I want to pose one question for you. Can you obtain 24-color, high-quality data from a 3-laser spectral flow cytometer? The answer, of course, is yes. So here's an example of a 24-color panel that the team here at Cytek came up with. And just to let you know, this wasn't something we could just whip together and try on the first try, get it right. We had to go through some careful panel design, thinking of how we were going to pair together the available dyes, seeing how the dyes looked on our system, and kind of thinking through, can we see these? Can we identify them? We'll be able to see the dim markers from the bright, and even iterate a few times to come up with this final 24-color panel. But with a little thought and a little effort, we got this one tube to show us these 24 different antigens, which I'm showing you here with a light wash sample prep of human whole blood. And as you can see, you can get a whole lot of information, all from one tube with a spectral cytometer with 24 different dyes combined. And not only that, you get excellent resolution of your negative populations and of your positives. Look at these TCR gamma deltas and how they're coming off of these CD3 cells. Or over here on the right upper corner, you have this great separation of the Tregs coming out. There's many examples here that you can walk through, but let's keep going. And if we have this high quality data, our spectral flow software is meant to acquire the data, but not to do deep analysis of your spectral data. Hence, pairing with de novo was a great choice, because look at some of the things you can do here. You can take your plot and gate out in FCS Express and run t-SNE analyses and do overlays like the CD3 overlay here. Over on the right, you've got lymphs, monos, and grams in different color codes. And you can subset and gate on these t-SNE plots and do overlays. Like these 6 down here are all showing the lymphocytes with different antigen markers overlaid on each. You can go even further, gate on a specific subset of interest and look at a bunch of different markers and how they're expressed on the cells in your gate. But Sean will get into all of this a lot later. So hopefully we've got you excited and interested, and I want to talk through how all of this is possible. But first, we've got to step back a little bit and think about where the world of flow cytometry has been going. So in the 1980s, one of the first commercial cytometers came out with a single laser and 2 dyes that you could use. And ever since then, roughly every decade, more lasers have been added, and slowly more dyes have been developed. So up to 2010, you could do a 3-laser system with 13 different colors coming from one sample preparation. However, all of these methods all use this, what we call conventional flow cytometer detection method, where you have one detector to pick up each of the dyes in your panel. Now compare that to what came out in 2017 when the Cytek Aurora launched with the spectral unmixing approach, Now you're able to use those same 3 lasers, but get as many as 20 colors or more as I showed you in that first slide. And the question our team would like to answer this year is what's possible when you have 5 lasers on board? And we hope to come back with answer to that question later this year. So how do we make this happen? Well, first, we have a very unique optical design. The schematic here on the right is showing you a 4-laser system, and all of the lasers are spatially separated as they enter the flow cell. As your sample is passing through, we have a forward scatter detector detecting the forward scatter light, and we have a dedicated module made of avalanche photodiodes, or APDs, for each of the lasers. Additionally, there's 2 side scatter detectors, one off the violet laser and one off the blue laser. And we take all of this information from APDs, which have a higher quantum efficiency than traditional PMT-based systems, and they're much smaller, so we can fit a whole lot more of them onto our system in much smaller footprint than a PMT-based system. So for this 4-laser system, we've got 48 different fluorescence detectors on board. And we take all of that information from every single module and stitch it together to get what we call a full spectrum. Let me walk you through how that works. For the violet laser, we have 16 detectors in that module, and each of those detectors has a filter in front of them, about 15 to 30 nanometer bandwidth for each of them, as shown here. And every time a particle passes through, we pick up signal in all of those detectors, and we keep all of it. We don't subtract it out from other detectors around it. Same thing for the blue array. There's 14 detectors on that module. And if you have a 4-laser system, the yellow-green module has 10 different detectors on board. And finally, the red has 8. So altogether, those 48 different detectors are stitched together with whatever signal comes through for your events to create this full spectrum diagram. What I'm showing you here on the y-axis is the fluorescence intensity, and on the x-axis are all of the different detector channels, starting with the violet, the lowest wavelengths first, denoted V1, V2, and so forth, then the blue, the yellow-green, and the red. Then what you're seeing in each one of these channels, for V1 for instance, you're looking down from the top of a histogram. So the red is the highest density number of events, the yellows have a medium density, and the greens and blues, there's fewer events falling at those MFIs. You take all this information and piece it together, and that's the full spectrum signature for this dye. The one I'm showing you here is for PE size 7, and for each of your controls, you get a unique signature like this to define your knowns on our system. So when you run your unknown multicolor sample, our unique spectral unmixing algorithm is going to go through and compare your mixture against all of these knowns you've defined with these full spectrums to figure out who's who and who's what in your sample. Now, with the power of this full spectrum unmixing tool, you as the user have a couple new superpowers. One of which is you can use new dyes in combination together that you probably haven't been able to use successfully in the past. For example, here's one where we took APC and Alexa Fluor 647. When you look at them on a traditional spectrum viewer online, they're very, very close. Their peak max emissions are nearly identical, and even the shapes of their emission spectra are quite similar. But through the eyes of Aurora, look at all their differences across our full spectrum signature. In the violets and the blues, the APC has some unique excitation that you don't see on the Alexa Fluor 647 in the plot on the bottom left. And then even in the yellow greens and the red, where they both have emission characteristics, the patterns you see are a little bit different on each. Because we can see these differences, certainly our unmixing algorithm can too. And here's an example of what that can look like. On the right is a simple 3-color panel with CD3 FITC, CD56 Alexa Fluor 647, and CD8 APC. We gated on the lymphocytes on the scatter plot, and then gated on the CD3 positive T cells and the non-T cells on the CD3 negative, and show both of those below on 8 versus 56. And you can see these population and subsets resolve very nicely with this spectral unmixing approach. Now you might be wondering, why choose these dyes that are so close together when I could just choose something else? And couldn't these combinations of really close, highly overlapping dyes introduce some artifacts into our data? Well, let's do a comparison. So for those same 2 dyes, that same combination, 3-color panel, if I take the CD56 and put it on PE, which is on a different laser, it's like our gold standard control on the top plot here, I gate those same cells, the NK cells on the top left and the NKT cells on the top right, and look at the percentages I'm getting. Roughly 28% on the NK cells and 17% over here on the NKT cells. Now, if I compare these to the plots on the bottom, which are the ones I showed you on the previous slide, but now I have the CD56 on Alexa Flora 647, let's look at the numbers. On the NK cells, I'm getting around 29% out of the parent, very similar to the 28 shown above. And then for the NKT cells, I'm seeing 17% parent, which is identical to what I was seeing when CD56 was on PE. The events are being classified correctly. Another superpower you have is the ability to extract the autofluorescence of your sample. Now, if you're running a simple panel with lymphocytes, there's not that much autofluorescence introduced, so you'll still be able to resolve dim markers on your violets and blues. But if you look at the spectrum on the bottom, this is a colon carcinoma cell line, you can see based on the MFI intensities in these violet and blue detectors that there's quite a bit of signal coming through. So if I have any violet dyes that are peaking in these lower violet channels or lower blue channels, I might have a hard time resolving them with this autofluorescence signature in the mix. But with the ability to extract that out, let's go through some examples of showing you what's possible. Here's one using the prime flow assay on Thermo Fisher. And it's a simple single color control, Alexa Flora 488, shown here on the bottom plot, and the unstained sample up above. We're only doing a single color staining, trying to decipher this 488 signal from the unstained, but they're quite similar. There's only a little bit of difference in the early blue channels between these two patterns that you're seeing here, these full spectrum signatures. Now, if we run it on our cytometer and look at the raw data, here I'm showing a histogram of B2, the second blue detector, where the Alexa Flora 48 has its maximum emission. And you can't really see a difference between the negative particles in the mix and the positive particles. Then if we unmix it without extracting AF, we have this plot here, where I'm now seeing Alexa Flora 488 on the bottom. Again, no separation between my negatives and my positives. Now, if I re-unmix the data and extract out that autofluorescence signature from the unstained sample, now I can clearly resolve what's positive and what's negative with that autofluorescence out of the way and not clouding my view of the data. Here's another example for a lice no wash assay using human whole blood. And on the left side, all the figures are with the data unmixed, having autofluorescence extracted. On the right side, we did not extract autofluorescence. So if you go from left to right and compare the green boxes, the purple boxes, and the blue boxes to one another, you can see that the appearance of the negative changes quite dramatically when you extract autofluorescence. Suddenly you're able to see where your positives and negatives separate out, and you can get on them way more easily than you can when you don't extract the autofluorescence from the sample for this particular prep method. So how do we get there? How do we generate all of this data on our spectral cytometers? I'd like to walk you through that workflow and show you how easy it is to get through the SpectraFlow software. So first you need your unstained control, and this is required in order to capture the sample's natural autofluorescent signature. Even if you choose to unmix and not extract it, we still need it there to account for it. Next, you need to prepare all of your reference controls, your single stains, and record those so that you generate these signatures for every single dye that's going to appear in your multicolor samples. Once you have that, you're going to go through our unmixing workflow. And when you unmix it, you're taking the data from what you see on the bottom left here, where you're looking at the raw data, which is the detector by detector data. So V1, V2, V3, et cetera, are displayed here. But once you unmix it, we're then able to assign the fluorochrome names to the data and look at it in the way that you're used to seeing it with your fluorochrome names on the axes instead of the raw detector names. Once you've unmixed it, you can then acquire your data in this unmixed mode and view it like this when you're recording your multicolor samples. Now I'd like to go through this workflow in a little more detail, but through our plate loader UI, since Cytek recently launched the plate loader. To use the plate loader, you need to have SpectraFlow version 2 software, and that software supports standard height 96 well plates on the loader in round, V, and flat bottom geometries. We've provided you with the flexibility to customize your loader settings and your acquisition settings. You also have the power to enable mixing one well at a time through a combination of paddle mixing with aspirate and dispense methods. Also, we've provided some predefined loader settings, which you can choose if you want to optimize for speed, doing a high throughput preparation, or if you want to optimize for low carryover and make sure nothing's getting from one well to the next. So to start things off, you create your new experiment. And you can see on the top, there's all these tabs that we're going to work through from left to right. The first one is defining your fluorescent tags. You have to tell the software all the dyes that are going to be present in your multicolor samples and define them here, where I'm showing I have 3 tags defined: 421, super bright 436, and BV510. Next, I go to the group section. I need to add a plate to the experiment. I have to choose if I want U bottom, V bottom, flat bottom, and I can drag and drop and select these different wells here, and I can add a group, a sample group, indicated by this folder with a plus icon, or I can add a reference group, which is the folder with the R on it, or I can add cleaning wells if I want to clean the probe in between some of my wells, this folder with the C on it. Once I have my groups identified, I can add markers and keywords. And I want to show you another view here from the acquisition pane, where if you need to go and change things later, you can use the same plate layout and drag and drop and add groups and change things up here as well. There's multiple ways you can modify your experiment in our SpectraFlow software. The markers page looks like this. If you want to add CD3, CD4, whatever you have going on your multicolor panel, you can add that here. In the next tab, you can create custom keywords and drag and drop those onto your tubes and wells and have those saved with your FCS file. Next, you need to define your acquisition settings, and you can do that at the plate level, the group level, or even at a well by well level. And the options you have are you can choose which worksheet you want to link to the tube when you're recording the data. You can set your stopping criteria based on event, time, or volume. And if you want, you can select a storage gate so that you're not saving every single event into your FCS file, and maybe you want to cut out some data and reduce the file size. Last but not least, you need to define your loader settings. And we provide 3 for you: the high throughput, low carryover, or default mode, but you're welcome to customize them. And the options you have are the acquisition direction, how long you're mixing and how fast you're mixing, how many sit flushes you want to do between wells, if you want to recover sample or not. You can set a stage temperature to cool down the stage a little bit, and you can preview your sample for a few seconds if you'd like by toggling that record data delay time to some number of seconds. Here's what those same loader settings and acquisition control menus look like when you're in the software running your experiment. So even if you're not in the wizard, you'll still have access to change things on the fly as you go. In the acquisition control pane on the right, you have the ability to toggle the flow rate, and there's 3 available to you: low, medium, and high. And for plate mode, low is at 15 microliters per minute, medium at 30, and high will run at 100 microliters per minute. So now we've got our experiment set up and we're ready to run. So we load our plate, and you can see in the bottom right what that looks like. And while we're recording, you're viewing the data on this raw data worksheet that I showed you before. And in this worksheet, I've got a gate on my lymphocytes, which is displayed on these nice spectrum plots in the worksheet. And as you can see in my lymphocyte population, I've got 2 different spectra going on here. I've got one from events expressing the positive BV421 signal, and I've got another beneath it, which is the natural autofluorescence in that unstained sample that's part of the mixture. In the bottom right, if you look at the loader, you can see the probe is moving back into a wash well. This is because I enabled mixing and washing as part of my run. So every time it's done recording the well, it moves to the wash well, does some sit flushes, and washes off the probe before it goes to the next well. Once you have all of your controls recorded, you then go through the unmixing workflow, where you identify for each control what the positive events are, what the negative events are, and then you can live unmix your samples going forward and view everything in that unmixed worksheet format shown here. Now, going forward, you have the ability to reuse your controls as well, so that you don't have to prep them every single day. But we advise that you try this and test out the different floors that you're working with, because we found that some floors are very robust over time, and some, the comps tend to change over time, and maybe they're best if we run them fresh with our experiment. You can kind of figure out what works for you and your panel. For the example I'm showing you here, we ran CD850, CD8E450, and CD8PE57, and we saved these signatures into the software in the reference control library on day 1. Then on subsequent days, we continued staining samples in the same sample prep method with the same antibodies, same lots and everything over time. And we record them as samples in subsequent days. Then we unmix those samples using the controls we recorded on day 1. And here's how things looked. You can see from day 1 to day 8, very little changed in the output of our cells here, our single stain cells, to day 14 and day 35. There's slight changes, but overall, we don't really need to continue to remake these controls every time we run them based on this data set. So to optimize your multicolor data and ensure that your outputs are as good as they can be, it often comes down to the quality of your controls. You want good inputs in order to have good outputs. So how do we achieve that and make these spillover calculations as accurate as possible? First, you want to make sure that you have nice, bright, positive particles, because you want to cover the full range of fluorescence that's going to be seen in your sample running through the system, so that our linear fit accurately captures that range. Next, you want to make sure the negative particles match the autofluorescence of your positive particles in each of your controls. So no mixing different bead types in your controls, because different beads have different autofluorescence characteristics, and the same goes for cells. You want to make sure you record enough events for your data points. If I only record a few hundred, that full spectrum signature is not going to look well defined. You're going to need a few thousand events. And if your marker is very dim, or if your control is a cell that's kind of rare, you may have to increase the number of events you record accordingly to define that spectrum. Last but not least, you want to make sure that the fluorescence spectrum of your control is identical to the multicolor sample. When you're using tandem dyes, you want to use the same lot on your control as you have in your multicolor sample, because those spectra could change from lot to lot for some of these tandems. Also, if you're doing something special to prep your multicolor sample, you want to treat your controls the same way, because your prep process can impact that fluorescent spectrum that you see. Now, in the software in 2.0, we have these great spectrum plots that you can throw on the worksheets, and they're amazing QC tools to check your controls. For example, here's a single stain bead control, and we're showing you gated on the 4th violet channel. You can see the positive and the negative events. And if we gate on the positives and show that in these spectrum plots, you can see this nice, clean spectra, the expected spectrum for this dye. Then if we display the negative events, you see a very low background. This is very characteristic autofluorescent signature of this bead. There's no other interfering dyes in the mix. So that's a good example. What happens when things don't go well? Here's one for Alexa Fluor 532, where on the top plot, you can see there's a lot of excitation in the violet compared to the expected spectrum beneath it. Turns out this user had a very fluorescent buffer in the mix that was creating all this really high intensity violet signature that they really need to wash out before running through their system and unmixing their multicolored data. Another example here is for eFluor 450. On the top, we're gating on these positive events and showing the positive bead signature. It looks great, nice and clean. You have the expected appearance in these low violet channels. However, when I move that positive gate onto the negative beads in the mix and look at this spectrum down below, there's something funny going on. It's almost like there's two separate spectrums. You can see the unstained autofluorescence signature, but then you see all this other highly positive stuff smearing in the mix. Turns out there was some APC contamination in that tube, so you have an APC spectrum mixed in with the autofluorescence, which is going to wreak havoc on their multicolor data when they unmix with this control. Let's recap everything we just talked through. First, Cytek's full spectrum cytometry gives you a whole array of new possibilities in your panel design. You have new combinations of dyes you can consider. You can get a lot more colors out of your data with fewer lasers, and you get really high quality data made possible by the APD detector arrays and our spectral unmixing approach. In the Spectral software, we give you an intuitive guided experiment workflow for both tube and plate acquisitions. And obviously getting good controls is critical to getting good multicolor data. You can use the spectrum plots to QC those controls to ensure that your multicolor data is the best it can be. And last but not least, the plate loader is now available. So if you're sick of loading tubes manually on your system, feel free to reach out to our sales team for more information on that. So now that we have our high quality data, what else can we do with it? And I'm going to hand it over to Sean.
Many thanks, Janelle. We're going to flip over the presentation to myself here. And again, if you have any questions for the Cytek team about anything you've seen so far, make sure to enter them in the questions and answers box. You've seen a few questions coming through already. We're going to be swinging back around to some of those at the end. But we're going to pick up where kind of Janelle left off. Once you have acquired your data using Cytek systems, well, now we have the ability to use FCS Express and work with the Cytek Spectra data files. And that's not simply the unmixed FCS, kind of standard FCS files. That actually means we can work with the spectral data as well as the raw FCS files together directly in FCS Express. So what we're going to focus on for about the next 20 to 25 minutes is using FCS Express with Cytek spectral data files. First, we're going to cover some of the important concepts for the file formats and loading that data into FCS Express and how to work with our new spectrum plots. How we can use spectral plots for quality control in FCS Express. So kind of picking up where Janelle left off on a lot of the topics she covered about quality control, we're going to take a look at some of those workflows in FCS Express. And then we're going to come back around and pull it all together. And how do you perform an analysis? How do you generate results and create high quality reports from those results in FCS Express? And as you probably know, you now have the ability to collect all of these new parameters and all these new combinations, which results in really high dimensional, highly multiparametric data sets. So we're going to cover at a very high level how to use tools like TISNI and VISNI in FCS Express to kind of help you distill some of those results. So let's jump right in here. We'll take a look at what these data files and what these formats look like. So what you're going to get when you export data from the Cytek instruments, or when you save that data, is essentially two data files. You're going to end up with a data file that has no suffix on here. You're going to end up with a data file that has a suffix of _unmixed. Now what these represent is one of these is the unmixed data file, the kind of standard FCS file that you'd use in any software, and the other file is the actual raw spectral data associated with that file. Now, when you're working with spectral data files in FCS Express from Cytek, you need both of these data files, and as long as they're both contained within the same folder for your experiment, you can load it into FCS Express and have access to the raw spectral data and have access to the standard FCS file data at all times. And to load this data into FCS Express, you can see that we have version 6 here running on the right-hand side, the latest release that includes this ability to read these spectral data files from Cytek. We can simply drag and drop. This is one way of getting data files into FCS Express. And when we're prompted by FCS Express for what type of plot would you like to visualize that data in, I'm going to choose a density plot, but I'm also going to choose spectrum. And that's going to load the data into one of our new spectrum plots in FCS Express. You can see how quickly that loaded up. And you can see here we have a full spectral plot, and we have the standard FCS file data coming in with that one simple drag and drop. Now, if you're not familiar with FCS Express, you can kind of tune in for some other webinars. You can contact our team, but the general idea in FCS Express is that it looks and feels like PowerPoint, so moving plots around the page. It works just like a PowerPoint presentation, and there's a lot of kind of easy clicks to get to different parameters. Like you can see here, if I want to change to side scatter and change to look at forward scatter, we can see that data. You'll also notice over here that when we're working with a spectral plot, we do have the option to click on the axis, and we can pick which part of the spectrum we want to look at, or we can pick the entire full spectrum here with this violet, blue, red A parameter. Now, when you create gates in FCS Express, you come to this gating tab and you just draw a gate around your population, and I'm going to draw a few gates here just for further visualization when we get into working with these plots. So just some simple gates as an example. And when you're working with these spectral plots, they have special options and formatting associated with them for visualizing this spectral data. Now, there's a lot of ways that you can format these plots, but if you simply double-click on a plot, it will open up the formatting window associated with the spectral plot. And you can see here you'll have options for the spectral plot. Now, our default display for these is a density mode, so color based on density. And there's a few options that you should be aware of. One is interpolate values, and this is essentially assigning some smoothing to this plot. So if I say set my interpolation to something like 4, you can see that plot becomes much easier to visualize. We've started some smoothing on that density plot to kind of help you better discern what's going on there with the spectrum. And additionally, if I want to do something like increase my resolution a little bit, we can actually further define that plot and look at that a little bit easier as well. Now, density plots are just one visualization of many for these spectral plots. If we choose this drop down here and say color on gated overlays, what we're going to see is that the colors that are associated with these gates over here on the left-hand plot are going to be backgated onto the spectral plot. And I'll also include a shade based on density, so that way we have backgating information and density information. So when I click OK here, you can see that the spectral characteristics for gate 2, which is in blue, are appearing in blue, and the spectral characteristics for gate 1, which is red, are appearing in red, and everything else is in black. And what you'll notice is that as I move these gates around in FCS Express, those spectral graphs update in real time so we can more easily visualize what's going on with these data sets back and forth between spectral plots and standard flow cytometry plots. But oftentimes it isn't necessary to view the entire spectra for every single event when you collect this type of data. So there's another option that we can access here in the Formatting tab. It's in this Overlays section, and it's what do we want to show in these spectral plots? Do we want to show all spectrums or simply the average of the spectra? And if we choose average and click OK, you can see that we're now viewing the average spectral line associated with events. And if we apply gate 2 by dragging and dropping onto the plot, we're now looking at the average spectra for gate 2 as we adjust that around. So you can use all of these kind of standard flow cytometry tools with these spectral plots just like you're used to doing with any other analysis in FCS Express. The additional functionality here is if you want to create a gate on a spectral plot and kind of grab one of the spectral lines, we can even apply that back over here to our plot. And if we arrange our gating hierarchy here correctly, what we'll be able to do is visualize the events back on that dot plot that fall within that spectral line. So the combination of using spectral plots and using standard flow cytometry plots is very, very easy to use in FCS Express. So you can try this out right away with the software, but what we'd like to do is show you how all of this kind of comes together in some of the workflows that Janelle had mentioned for quality control. So we're going to close this out, and we're going to take a look at one of our first examples here. And one of the first examples we're going to look at is comparing different dyes and looking at dye compatibility within a panel design. So what we have shown here is that on the left-hand side, we have a sample stained with Alexafluor 647, and we're showing the spectral graph for that. And on the right-hand side, we have a sample stained with Bio 667 and the spectral graph for that. And what we've done here is plotted an overlay of these two spectral lines for the two different dyes that are in the system. So what you'll see is when we look at this, they're very similar. We look at the overlays, they're almost completely overlapping. And really what this means to you is that this is one particular dye combination that you can't use in this system. But if we use our data list here on the right and we simply click on the second set of data, it's going to load in a new set of data, and this combination is BV421 on the left and eFluor 450 on the right. And this is a dye combination that you probably wouldn't be able to use in a classic cytometer. But if you overlay these spectrum, what you're going to see is that the majority of the spectrum looks the same, except for right down here on the low end. And we're going to just magnify in here and take a look at that on this chart. And this little dip in the blue line for the eFluor 450, this is enough differentiation between these two dyes to be able to use them in the same system and use them in the same kind of panel design. And again, getting to this information and using these spectral plot overlays and using this kind of technology in FCS Express makes it very easy to facilitate making decisions about what you can and cannot use in your panel design. So again, keep in mind here we've just loaded two sets of data. If you have a lot of different dye combinations, you can simply load them into the data list, quickly switch back and forth, and get you the information that you need to better move forward with your assay design. Now, another consideration that you need to make is when you're working with this data, what kind of controls are you going to be using? And are you going to be using beads? Are you going to be using cells? And as Janelle pointed out earlier in the webinar, that having these controls is very important, and you need the kind of right controls to compare apples to apples. So what we've done here in this kind of example is we have a sample for beads up here at the top row. We have a scatter plot, we have the spectral plot for that, and then we have a histogram here with the blue being the beads sample. And on the bottom here we have a cells-based sample, the spectral plot for that, as well as the histogram. Now, what's really important about this and working with this data in FCS Express is that by using gates, we can kind of clean up these spectral plots so we can better visualize and make sure that we're using the correct populations for our controls. So one way to apply a gate is we can take this beads gate here on this chart and apply it directly to the right here on this spectral plot. And we can see that that begins to clean up this analysis. We're removing a lot of the noise and a lot of the debris from this. So we probably want to do something similar down here for the cells gate. And you can see that when we're working with these gates in FCS Express, we have different shaped gates, but we're using the same gating hierarchy. This is called data-specific gates, where we can assign different gating positions for the same gate across many samples. And again, what we have here is you can see that this is starting to get cleaned up a little bit more. But if we really want to focus on the positive portion of this data, what you would find underneath this marker here, what we can do is simply apply this V14 positive gate up here, again through a drag and drop, and through a drag and drop. And we can see now that the spectral characteristics for the positive populations that are also falling within those scatter gates are very similar. And if we adjust this marker, we can get that information updating for us in real time, so we can make some good decisions about this. And again, if we take that overlay and we simply apply the V14 positive gate here, we can see how well the beads versus cells correlate. Now, again, there's a lot of different ways to visualize and display this data in FCS Express, and that's one of the more powerful tools about using FCS Express for these types of data sets, is that you can visualize these in a number of ways. So again, we have the same type of situation where the beads are up here at the top, the cells are down here at the bottom, but we're using those color density overlays here in these charts, where we're looking at all the events in blue, we're looking at cells within the scatter gate. In red, we're looking at cells that fall within that V14 positive in purple. And when you put these on a black background, the charts look really sharp, of course. But again, we can kind of come in here, start looking through this data and drilling down to, scatter versus positive. We can clearly see the distinction here between these populations, and we can drill down further into that. So again, using these visualizations in FCS Express is a really great way to kind of drill down into this data at the most basic level, making sure that you're using the correct populations and that you're using the correct controls when defining your experiments and setting up your assays. Now, there's a further consideration. Now that you've decided what your controls are going to be, beads versus cells, what your dye combinations are going to be, what you'd probably want to do is come in and figure out, well, which dyes am I going to use for which markers? And can that be done interchangeably? Can that be done reproducibly? And the answer is yes, it can be done interchangeably and it can be done reproducibly. And what we've put together here is an example of different dye combinations. So on this page, we're looking at CD4PE versus CD8APC. And we've done this using some data from the new plate loading system from the Cytek system. And you can see here that we've actually acquired samples in triplicate, where we're looking at one sample from well C4, again CD3 staining, and then our CD4-CD8 combination. And we've done this for 3 different wells. Now, in the next page here of this analysis, we've swapped out CD8APC for CD8A532. And again, we're making the same type of comparison here and seeing what happens with our gated populations for CD8 across all of these markers. In the next set, again, we're replacing CD4PE with APC and CD8 with PE Sci5. And you can see, again, we've done this with another set of dyes. So, this is a lot of information to distill, and one of the really nice things about using FCS Express is that we can simply graph and chart this data in FCS Express directly, so you don't have to take this and go out to Excel and go out to Prism. So what you'll see in this chart is we're plotting the average CD8% gated for our different dye combinations. So looking at PE versus APC, PE A532, right? We've switched out what is CD8 in all of these. And the standard deviations here represent the percent gated from each of the triplicates. And what you'll also notice at the end here is that we're showing the average for all 4 of these different combinations and the standard deviations between them. So really what the take-home message here is that you can use any of these dye combinations, and internally they're reproducible, and across all of the dye combinations they're reproducible. Now, again, we're charting all of this in FCS Express, and we're using what we call integrated spreadsheets, which you can see down here. It looks like a spreadsheet, just like you would use in Excel. And what that means is this is all linked to the data. So if I decide that I need to change my scatter gate a little bit and see how that impacts the results, you'll see these graphs changing, you'll see the standard deviations changing, all in real time. So you don't have to change a gate, re-export your results, recreate your graphs, right? And it doesn't just apply for doing things like this. Really, any sort of analysis you can do, you can use these live updating gates. And if we wanted to examine what happens when we move between the different populations, we get that final result in real time directly in FCS Express, and we can link it all back to the FCS file data very simply and easily. So once we've put all of this together, we've decided what our controls are going to be, beads versus cells. We've decided what type of dyes to use in combination, which types of dyes to use on which of our markers. We can put it all together and start an actual analysis for our data. So what we're going to do is show you what that can look like in FCS Express. And you can see here that we've built up an analysis. An analysis in FCS Express works just like PowerPoint, as I mentioned. And these plots are built up simply by inserting whatever type of chart that you'd like: histograms, spectral charts, dot plots, density plots. You draw the chart on the page just like you would insert an object in PowerPoint, and then you can switch to your parameters of interest and get analyzing with that. But some of the additional benefits of working with FCS Express is that any of your reportables, any of your percent gateds, any sort of kind of MFIs, custom statistics, they can be reported anywhere in the software. And you see that as I move this gate, all of our plots update, and those reportables are updating in real time as I move that around. And again, this is happening not just on one page, right? If you're working with, in this case, it's a 16-color experiment, you're working with these lots and lots of different parameters. You probably want to include many pages for this report and build up a kind of comprehensive analysis and a report that you can work with and manage that data very easily. Additionally, you see here that we built in a spectral graph as well, right? So instead of just using these standard plots, if I wanted to say, all right, well, let's look at our CD3 negative overlay on our graph of side scatter versus CD3, we can actually come in here and set some of our CD3 positive and negative controls based off of these spectral graphs. So kind of using an internal control of the system rather than using some sort of FMO, right? So there's a lot of different ways that you can integrate the spectral charts into your kind of standard analysis and your daily analysis for working with these data files. And what we're going to do here is apply this lymphocytes gate back to the chart. We'll get that over here and continue on with our analysis. Now, when we're working with these data files, in this case, we were only working with one data file. You can see it was a whole blood donor over here on the side. Generally, when you're analyzing data, you don't have one data file. You might have 3, you might have 30, you might have 3,000. Right? So if you want to use this as kind of a template for your analysis for all of your other data files, you can simply grab the data files that you want to analyze. You can drag them into the data list. They will appear there as kind of placeholders for those data. And if we click on the next, what we call an iteration or the next file, everything in the analysis will skip to the next data file, we can skip to the next one, and we can start looking all this data side by side and consolidating it and getting all the results kind of up front and ready for us to review and publish and do whatever we're going to do with it. But additionally, if you're working with many data files, just visualizing it is one thing, but getting this reported is another. So FCS Express has a lot of ways that you can export results. If we want to export what's on that page to a PDF or a PowerPoint or a 600 DPI image, we can do that. But also, if you want to quickly crunch through many data files, we recommend taking a look at some of our batch actions. And what we can do here is define a series of actions. Here we've defined export to PowerPoint, export some statistics to Excel. We can build these up, and building up the reportables to export to Excel, it's as simple as a drag and drop. If we want to put that % gated for monocytes, you can see it got added down here at the end of the list. And what will happen is when we click run on here, it's going to export to PowerPoint, it's going to export to Excel, and it's going to do that for as many different iterations as we have in our data list. So again, here we have 3. If you had 30 or 3,000, the most I've ever seen in the data list is something like 30,000 from a high content screen. You can crunch through all that data very quickly and easily and have it collated to tools like PowerPoint and Excel. And what you're going to see here, Excel is launching up with all of our reportables, including our keywords. We never had to type anything in. We never had to copy and paste. We have this integrated report. And additionally, when we come over here and we look at our PowerPoint, again, we're in PowerPoint now and not FCS Express, we have graphics in here, and they're also very high quality, which is very important. When you're getting ready to publish, you don't want to have to be moving back and forth between Photoshop and Illustrator, right? So these are high quality reports, and importantly, they're all vector graphics. And what that means is I can move these charts out, I can copy and paste them directly into another PowerPoint presentation. And even if I want to, if I'm working on Microsoft PowerPoint on a PC, importantly, you can ungroup these things. This is a pretty cool feature. And if you didn't want to change your axis label in FCS Express, which you could do very easily, you wanted to change it to FTSE CD3 here, you can change it. If you needed to grab something in, like the distribution of this data as part of your presentation, you can grab that. If all you wanted to do was kind of come in here and grab these spectral lines as part of your presentation, right? You can pull these things out, and it really facilitates getting to a publication-ready figure, some sort of PowerPoint presentation for your lab, reports and things like that very easily. So again, reporting all of this data becomes very simple. It becomes a very trivial type of thing to do in FCS Express, but you have all these kind of high-powered flow cytometry tools that we've been developing over the last 20 years. Now, when you're working with this data, the other thing to keep in mind is when we click on the parameter list here, it's highly multiparametric, right? We've got 16 colors coming from this particular experiment. One of the examples that Janelle showed in the beginning was coming from a 24-color experiment. So how do we handle that, right? We do have all the ability to work with these kind of classical flow cytometry style reports, but importantly in FCS Express, we offer the ability to work with high-dimensional data reduction tools such as TISNI, VISNI, and SPADE. So we're going to take a quick look at how those work in FCS Express, not in a lot of detail, but these are all accessible in our Tools tab under Transformations. And when you expand the transformations window, you'll see that we have the ability to perform a number of transformations, and we've natively built in TISNI and SPADE. And when I say natively built in, these do not rely on R. You do not have to be a programmer or a script writer or an biostatistician to use these tools. You simply select TISNI, you choose the file that you want to perform that TISNI on, you choose all of the parameters that you want to include. When you run that TISNI algorithm, here we'll just choose a few, and then you pick and choose from the variables associated with that algorithm. Now, when we're ready to run this TISNI, I'm going to click Calculate TISNI. By default, these things will be automatically calculated. But what you're going to see is when this TISNI is done running on this data set, my TISNI charts are going to appear here on the left the way that I have these set up. I can get this out of the way, and we can start analyzing this data or exploring this data with some of these tools. So if we wanted to come in and say, all right, let's look at the heat of CD4 on our t-SNE charts, or let's look at the heat of, something like our CD8 here, we can start using these t-SNE visualizations to kind of drill down into these clusters and these continents and help figure out what's going on. And again, this is all linked to the raw FCS files and the spectral data. If I wanted to take kind of one of my classical CD4 versus CD3 charts and apply a gate back onto my t-SNE chart and kind of get an idea of what these clusters are, again, you have that real-time gating and updating capability. What you'll also see on the left or on the right-hand side here is that we've backgated some of these t-SNE gates. So as we move this gate around, we can see where on that t-SNE chart these gated populations fall. And of course, you can take these gates and move them back to our classical flow cytometry plots. So if I wanted to drill down in this t-SNE chart and look at the CD3 versus CD4 phenotype and the CD4 versus CD8 phenotype of a cluster, we can drill down and get that information very quickly and easily and report that all very nicely in FCS Express. So even though you're working with very high dimensional data, you can work with those kind of templates for analysis, you can build them up and you can reuse them. You can go into further kind of data exploration tools like t-SNE and SPADE. And again, we make these very easy to use. We make them very accessible to any level of user, where you simply come in, you check the boxes that you want to perform the cluster analysis on, choose the variables, and run that. So with that, we're coming towards the end of the presentation here today. And what I wanted to do is summarize some of the items that we discussed in regards to FCS Express today. And importantly, FCS Express allows you to easily access the raw spectral data and link that up with your standard flow cytometry data so you can work to generate reports and quality control once you come off the instrument very quickly in FCS Express. And as you've seen, those spectral plots may be adjusted and formatted to your experimental specific and publication requirements very quickly and easily. Generally just a right click and format or double click on the plot. You can change anything about those plots that you want, whether it be the title, the spectral average, what you're plotting, the axis labels, all of that can be adjusted. And importantly, integrating the spectral plots can allow you to assess, optimize, and perform this type of quality control directly in FCS Express. And these analysis layouts, once you're done with your quality controls, can be set up easily, just like creating a PowerPoint presentation, and used for many data files in a batch process and saved for reuse. So you don't have to recreate the wheel every time you come back with a new 16-color or 24-color experiment. You can just use something that was similar that you did last time. And again, very importantly for these high-dimensional or highly multiparametric data sets is that the data reduction algorithms and tools in FCS Express are easy to use. They help support this highly multiparametric analysis from the Cytek instruments, and they're very accessible to every researcher. Again, you don't have to have some sort of PhD in writing R scripts to do that in FCS Express. So again, what we'd like to do is thank you for attending the webinar. And thank you very much to Cytek Biosciences and all the support that they've given us over here at Novo Software to help integrating this technology directly with FCS Express and bringing that to all the users in the system. And again, thank you very much for joining us today. We're going to be opening up the floor for a few questions here. And Janelle, thank you very much for your presentation today as well.